Hello. So I'm pleased to welcome you to the webinar today organised by the Biochemical Society and Portland Press. And this is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for further details. My name is Professor Jennifer Pocock, and I am Professor of Cellular Neuroscience in the Department of Neuroinflammation at University College London. And today's webinar is entitled Exploring the Role of Microglia and Astrocytes in Neurological Disorders, and is part of our dedicated early career research programs of webinars. We are delighted to provide this opportunity today for Raul and Annie to share their work with the molecular biosciences community. Before I hand over to our first speaker, I'd like to mention that questions will be asked at the end of the webinar, but please do send in your questions during the talks. If you have a question, please type in the question box as shown in the image on screen, stating who your question is for, and we will try to answer as many as possible at the end of the seminars. Our first invited speaker today is Raul Loera-Valencia from Tecnologico de Monterrey, Mexico. Raul is a research professor at the School of Medicine and Health Sciences. He completed his master's degree in molecular biology and his doctoral degree in molecular biology at the Institute of Scientific and Technological Research of San Luis Potosi. He also conducted postdoctoral research at McMaster's University in Canada and subsequently pursued a 6-year postdoctoral research position at the Karolinska Institute of Stockholm, Sweden, specializing in dementia research. So please, Raul, give us your presentation.
Thank you, Jennifer, for a nice introduction. Can you hear me all right?
Yeah.
Perfect. So good afternoon and good morning to all the attendees. Thank you for being here. So I'll just start. I'll just reset the clock here. So my talk is entitled Synaptic Dysfunction Mechanisms from Oxysterol Imbalance in Astrocytes. And I will tell you about this work that was done mostly at Karolinska Institutet in Stockholm, Sweden. And then after that, I came to Mexico to start my lab in the Tecnologico de Monterrey. So that's where the work has been done. So let us begin by discussing a little bit about the role of cholesterol in Alzheimer's disease. I mean, there are several papers that have come up, and I'm just showing you one result for a study with 2,000 patients with early onset Alzheimer's disease, where they studied the levels of cholesterol in serum in the form of LDL in the plasma, and then we can see a positive correlation where patients with Alzheimer's have elevated cholesterol. So there was an association between these two levels. However, cholesterol do not cross the blood-brain barrier. So how does cholesterol can be related to the pathology of Alzheimer's disease? Well, the main mechanism that we are studying is a mechanism in which cholesterol is metabolized by cells in the peripheral tissues through this enzyme CYP27A1, which produces 27-hydroxycholesterol. So we call this metabolite 27OH, right, or 27OHC. So then this is able to cross the blood-brain barrier and enter into the brain, where it's further metabolized by neurons. So the neurons, they have this enzyme CYP7B1, and then they turn it into a bile acid intermediate that can cross the blood-brain barrier and then becomes excreted afterwards. However, in pathological conditions, when we have hypercholesterolemia, we have elevated levels of 27-OH, and that goes into the brain and then surpasses the capacity of the neurons to metabolize it. And then it starts to do damage to the neurons, to the microglia, and to the astrocytes. However, this was not very well characterized when we started this research. So this came up. Sorry about that. Yeah. So we started doing a culture, primary culture of neurons, and then we treated with 27-OH 1 micromolar. So that's about 3 times the physiological level. So it's not super high, but it will be equivalent to hypercholesterolemia. And then you can see that we have decreased arborization in the dendrites. And also we had very severe effects on PSD95. And also we knew that there were alterations in this transcription factor called REST and PTBP1. Also we had a decrease in microRNAs that were proneuronal. So long story short, we deducted, or we found out that RXR gamma was a receptor that was involved, because whenever we did small interference RNA, it would partially rescue the phenotype, right? And then we came up with this cascade where 27 was dimerizing with RXR gamma, and then inducing these, you could say, antineural genes that were affecting our neurons. So when we tested brain slices from the transgenic mouse, we found quite interesting result with the LTP. We did the long-term potentiation, and we saw kind of a feature here where you have increased LTP, but this didn't mean that the mouse was learning better, but we interpret this as an aberrant form of LTP, right? Like this is deviating for the wild type behavior, right? And it was kind of like giving us the impression that there was some kind of hyperexcitability phenotype. So then we look at the glutamate transporters in the hippocampus, and then we saw no difference, and we saw no overexpression in the glutamate receptor levels in the transgenic mouse. So that was strange. So this is why we came up with an alternative hypothesis for this, where we were thinking about astrocytes, right? So what would happen if 27-OH levels that were elevated were impairing astrocyte synaptic function? And then that meant it was somehow inhibiting the recapture of glutamate from the synapse. And then if we have a downregulation on the glutamate transporters, then we will have like an increase in the glutamate levels in the synaptic cleft, right? And of course, also maybe it was affecting the energy supply as well. So then this was our new hypothesis to try to complement what was happening in the transgenic mouse. So we first looked at tissue that we had from mouse that was under a high fat and high cholesterol diet for 9 months, and then we checked the hippocampal lysates, and we did find some astrogliosis. When we looked at the FAP expression, it was increased, but we didn't see much in terms of the glutamate transporters. However, when we checked the CYP27 transgenic mouse, this is hippocampus and this is cortex, we did see GFAP increased, and together with the downregulation of the main glutamate transporters, GLT1 and GLAST, right? And the same happened, at least for GLT1 in the cortex, although GLAST was not significantly different. Oh, so okay, so we knew that this was happening. There was something happening here that was relating to the phenotype of hyperexcitability. So we did some cultures, primary cultures of astrocytes in 2D, and to our surprise, there was not much happening with the 27-OH. So we even tried incremental concentration, and we went up to 10 micromolar of 27-OH, and the astrocytes didn't seem to care too much about it. There was no stellation, there was no decrease in cellular abundance, However, whenever we took the induced media and put it on differentiating neurons, we did see again the downregulation of PSD95. And this was not the role of 27OH, because 27OH gets metabolized very quickly in about 1 hour. So this was a media that had been in the astrocytes for about 6 days, right? So then there was no 27. However, something in the media was inducing PSD95 to be downregulated in the neurons. So this told us about the crosstalk between cell types in the culture, right? So that's why we came up with the idea to do a co-culture, and we decided to do it in a 3D configuration, because we wanted 2 things. We wanted the first thing to be more resemblant of the physiological conditions, and the other thing that we wanted as well to do is not having any fetal bovine serum exposure. So we were using a defined media for this, and this was a 3D culture that was based on microglia. And it contained the cell suspension from embryonic brains from day 16 and no more than day 19, but mostly we did brains with 16 days. And then we were able to have a nice co-culture where you can see here the neurons in green and the astrocytes in red. And we acquired this with confocal microscopy. And we know that at least in our cultures, we have neurons, astrocytes, oligodendrocytes, and microglia. But for this study, I will just talk about the analysis of neurons and astrocytes. So we did the analysis using IMARIS 3D reconstruction. So with IMARIS, you can take the astrocytes, and you can also map the complexity of the neurons and do several types of morphological analysis. So it will do so by doing surface rendering. So whatever image we have from the confocal, IMARIS will transform into a 3D render that we can then analyze, right? So, all right, so then when we treated the astrocytes in 3D in co-culture with just 1 micromolar 27OH, we did see now an effect. And now we saw this severe effect on the astrocytes, where they were having less branching, less complexity, and you can see here the quantification in the Scholl analysis. 27OH treatments were diminishing all the complexity of the astrocytes, right? And this is also quantified in Scholl intersections, and also have diminished length. And we actually could see the populations changing with the treatment, right? So 27OH would give us more smaller and less branched astrocytes graph here. So then this was not happening at all into the cultures. So when we look at the gene expression of the whole culture, we could see again the downregulation of PSD95. And we saw again what we were looking at in the CYP27 transgenic animals. We have GFAP increased and GLT1 and GLAS downregulated. And again, as the first paper I showed you, we saw a decrease in ARC expression. And in this case, we didn't have a significant increase in LXR beta, but we did see a change in expression in RXR gamma at 3 days in vitro. However, this was downregulated instead of being up, right? So, okay, so we knew that this was happening according to our hypothesis, but we wanted to test if this had any impact on the excitability of the neuron. So we did, in collaboration with Luis Arroyo and with Hugo Valleza in Karolinska, we did electrophysiological recordings on the 3D culture. So you can see here one of the neurons treated with 27, and what we saw was an increase in the excitability of the neuron. So you can see here the number of action potentials were increased, and as well you can see here how whenever we apply this current, we will get more action potentials from the neurons, right? And also, when we analyze the spontaneous firing, we will have more in the 27OH. And also, whenever we were checking the connectivity to see, let's say here we were listening to the neuron to see the input that was getting from the network, and then you can see here the excitatory potentials that they were more frequent in the 27OH treatment compared with the vehicle and with the non-treated control. So then that kind of gave us the confirmation that we needed that this there was something happening where 27 was affecting the glutamate transporters, and then this was giving the neurons a phenotype of hyperexcitability. And those are our take messages for home, is that we do see that the 3D co-culture recapitulates many of the features of the transgenic animal, and we have a unique response from the astrocytes that is not seen in 2D configurations, and then 27OH affects the astrocytes, and that gives persistent hyperexcitability to the neurons, which is something that is seen a lot in Alzheimer's disease and other neurodegenerative diseases where you see hyperexcitability in the neurons. And I just want to thank, of course, the leaders of the lab in Karolinska, Silvia Maioli and Angel Cedazo. Of course, I did not do this work alone. We had a big team first and then a smaller team, but I had help from very talented people like Mr. Spanos here, and Maria Latorre, which was my PhD student, and Julen, who is now doing a very nice postdoc in the United States, and Maria as well. And of course, to thank the funding for the private organizations in Sweden that supported our research. So thank you very much.
Sorry, I've got to share my webcam. Thank you very much, Raul. Well, as I said, we'll wait to the end to get the questions. So our second speaker is Annie Ciernia from the University of British Columbia. And so Annie is Associate Professor and currently the Tier 2 Canada Research Chair in Understanding Gene Expression in the Brain. She started her lab in the Department of Biochemistry and Molecular Biology and the Javid Moafagi and the Center for Brain Health in 2019. Annie's research interest is in the understanding of epigenetic mechanisms of transcriptional regulation involved in brain plasticity and neurodevelopment disorders. Her lab's current research focuses on understanding how early life perturbations to the epigenome alter microglial neuron interactions in the developing brain and lead to lifelong changes in cellular function and behaviors. It's all yours, Annie.
Great, thank you so much. You can hear me okay?
Yeah.
Terrific. Okay, so thank you very much to the organizers for allowing me to share my lab's work today. So I'm going to tell you about a new unpublished story that my group has been working on. And there we go. So my lab is located at the University of British Columbia, and I just want to recognize that we work on the traditional, ancestral, and unceded territories of the Musqueam people. And it is really our privilege to live, work, and play in their backyard. So today I'm going to tell you a story about how microglia regulate gene expression during brain inflammation. So what are microglia? So microglia are the innate immune cells of the brain, and they're shown here in the mouse hippocampus, where each golden star structure is an individual microglia, and you can see they nicely tile across the entire brain, and they make up about 10% of all brain cells. And as the resident immune cell in the brain, they are incredibly important for fighting off disease and injury and a whole host of other homeostatic functions. So my lab is interested in understanding how microglia are regulated in the context of inflammation. And we are using a commonly used model in the lab to induce microglial inflammation. And we do this in mice by treating them with lipopolysaccharide, which is from gram-negative bacteria, so something like E. coli. And we can inject this peripherally into mice, and we can see how the microglia respond in the brain. And so what you can see is that microglia rapidly shift from this nice homeostatic state where they have all of these lovely branches that are surveying the local brain environment. And if you add LPS, they then shift to an inflamed state. And we can see this in a number of different measurements, including increases in pro-inflammatory gene expression and changes in the microglia's cellular shape. So their morphology changes to be less branched and more rounded amoeboid. And we like this model because it's easy to reproduce. All of my students can do this pretty much on the first try, and it's time locked. So we know exactly when that LPS was given, so we can look at how the microglia are responding. And my lab is interested in trying to understand a phenomenon called microglial memory. And to explain this, if we look at the responses of microglia, so you can measure a number of different changes in the immune system to a number of different stimuli. So microglia will respond to infections, pesticide exposure, stress, other types of inflammation conditions, and they will induce pro-inflammatory gene expression, change their morphology, release a bunch of cytokines, and try to basically ward off any injury or damage happening in the brain. Once the actual stimulation is over, microglia return to their homeostatic state. But what we're interested in is then what happens if you give a secondary stimulus. And there are a number of different things that have been characterized, including two forms of what we call microglial memory. And so this secondary stimulus can induce something called primed immunity or training, where you get an exaggerated response in the microglia. And so they hyper respond. And this happens even if the second stimulus is the same as the first one. And there's the flip side of this, in which the secondary stimulus produces a suppressed response. So the microglia tolerize, and they do not respond at all or very little to that secondary stimulus. And both of these phenomena have been observed in the literature, but we don't really know what the state change is in microglia that underlie this altered response, and if this either predisposes or protects to subsequent disease susceptibility. And so we're interested in this immune memory phenotype, and if we could potentially understand how that works. And so our hypothesis that we're working on is that there is a gene expression or an epigenetic mechanism that controls the switch between microglial priming and tolerance. And this is important because we could potentially manipulate this response to mitigate damage of neuroinflammation and disease. And so there's been a fair bit of literature looking at different types of paradigms to study microglial immune memory. One of the first ones was to look at a low dose of LPS in an Alzheimer's or stroke model, and low doses of LPS produced training or priming, so hyper-responsing. And if you gave multiple doses of LPS, sort of cumulatively over a short period of time, you could produce tolerance. And then this was followed up by another group that showed a single higher dose of LPS produced tolerance to LPS up to a month later. And then people have looked at a number of other different types of primary stimulation events, including things like maternal high-fat diet, followed by LPS, which can produce priming, or poly-IC, which is a viral mimic given very early in life, so on embryonic day like 9.5, and then stimulate the animals with LPS as adults, and that can produce tolerance. So it isn't quite clear what the rules are about inducing priming versus tolerance, and which types of stimuli are inducing which type of effect. And so when we started doing some of this work, my PhD student Morgan Taurus developed a paradigm in which we can give a medium high dose of LPS or vehicle, which is phosphate buffered saline or PBS. We can wait 7 days, and then we can give a second injection of LPS or vehicle. And the first indication that this might be a good model for tolerance was when we looked at the sickness behaviors of the animals. And so just to orient you, the higher the score, the more sickness the animal demonstrates. And we have, in this case, our vehicle control, where you get almost no sickness. If we give LPS and then 7 days later a vehicle, we get basically no residual sickness left over after that week. If we give vehicle and then LPS, we induce the acute sickness response as expected. But if we give LPS twice, we actually get a really nice blunting of this sickness response, very consistent with tolerance. And so what Morgan did was then collect microglia 3 hours after this second injection, and then we did RNA sequencing on them in both males and females. And so the first thing we wanted to ask is, does gene expression return to baseline by 7 days post LPS? And this is important if we want to look at cumulative effects of 2 doses of LPS. And so the answer to this is yes. So after 7 days, there's no difference between having LPS 7 days ago and just having vehicle. So that was good. We then asked, does gene expression actually increase in microglia in response to LPS? And this had been pretty well demonstrated in the literature. And we were able to nicely recapitulate that. So now we're comparing vehicle versus vehicle LPS. And we get a whole bunch of genes that change. So the acute LPS injection increases about 3,000 genes in both males and females. And shown by the Venn diagram, many of these genes are shared across the 2 sexes. And we also get a similar number of genes that decrease. Again, the majority of which are shared across the 2 sexes. And so this was a good indication that our model was working as expected. So then we wanted to go in and ask the question, what actually happens in this memory condition where you have the 2 doses of LPS? And the first thing we wanted to do was to compare these 2 doses of LPS to both of our control conditions. So to having LPS 7 days ago or having just vehicle. And so just to orient you to what this looks like, so we're looking first at genes that increase in response to 2 doses of LPS. So we have our 2 doses of LPS that are greater than 1 dose of LPS 7 days ago or greater than PBS twice. And we get about 1,600 genes at the center of this Venn diagram that indicate we're seeing real increases just to that 2x LPS. And we see about 1,000 that go in the other direction. And so then we took these genes and asked, is there an effect of having prior LPS on the effect of the second LPS? So does the 2x LPS matter? Or is it just the same as having a single LPS? And it turns out it makes a fair bit of difference. So we have several hundred genes in females and about 150 in males that increase specifically in response to 2x LPS over and above a single dose of LPS. And similarly, we have a response of a suppressed gene expression in response to 2x LPS compared to a single dose in both the males and the females. And you can see we have some sex differences in this. So we then went ahead and took a look at what these genes actually were. So I'm going to show you a big heat map, but just to orient you. So we clustered our data. This is unbiased hierarchical clustering. We do not see any effect of the date that we actually collected the samples, which was good. If we look at sex, we actually have nice separation by males and females. So females are purple, males are blue. We have great separation by that second dose of LPS. And interestingly, we have some separation by the first dose of LPS, but only in the condition where they also got a second LPS dose, indicating that the first dose of LPS is really modulating the response to this second dose. And you can see that probably more clearly when we actually look at this heat map. So in this, each column is a sample and each row is a differentially expressed gene. So I'm going to walk you through each one of these. So the first category is genes that are lower in expression when you have 2 doses of LPS compared to 1. So these are things that are suppressed in both males and females. And the suppressed genes are involved in cytokine signaling, immune responses, and generally pro-inflammatory genes. And we took a look at the promoters of these genes to identify potential transcription factors that might regulate this specific set of genes. And you can do that by looking at the DNA within the promoter region. And we have significant enrichment for motifs for NF-kappa B and IRF1, both of which are known regulators of pro-inflammatory gene expression. So this was exciting. It indicates that we really do have something that looks a lot like microglial tolerance, where we can suppress the induction of pro-inflammatory gene expression. And to give you an example of what this looks like, so this is our 4 groups again, our 2 control conditions, which are relatively low. So this is, for example, 3 pro-inflammatory cytokines. We get nice induction with a single dose of LPS, and that's suppressed if you have a prior history of LPS. And this is true for all 3 of these cytokines, as well as several of the subunits of NF-kappa B and other types of regulatory immune factors. And we actually have now gone on and validated this by qPCR in a completely independent set of samples. So this is for IL-1 beta and TNF-alpha, 2 pro-inflammatory cytokines, where we can get nice suppression in our tolerance condition. So these are genes that are happening. This is happening in both males and females. We then took a look at our female-specific genes. And again, we see many of the same types of genes. So these are pro-inflammatory regulators, things involved in cytokine production, and TLR4 signaling. So TLR4 is the receptor for LPS. And we looked at the promoters of these genes and again saw similar types of regulatory factors. So these are all known factors that are involved in regulating pro-inflammatory gene expression. So to give you an idea about what these look like, so for example, here's MyD88, which is downstream of the TLR4 gene, and that's suppressed in both, in this case, more strongly in the females, and then also STAT3. Interestingly, we do not see changes in the TLR4 receptor expression itself. It goes down in response to LPS regardless of history. So we have some interesting things that we can look at now in regard to the actual signaling response to repeated LPS. And then if we look at the male-specific genes, there's about 70 of these, and they're involved more in like kinase activity and other types of transcription regulation. We have a number of them that go down specifically only in the males, including a number of signaling molecules. We did not see any transcription factor enrichment in those ones. And we also have a bunch of things to look at in the genes that go up. I don't have time to show you them today, but I'm happy to answer any questions about them if people are interested. And so we've also looked to see if these effects extend beyond 7 days. So we did a 30-day spacing with the same paradigm. And again, we see very similar suppression of the sickness response to 2x LPS. We see similar suppression of gene expression to IL-1 beta and TNF-alpha. So indicating that this is really a long-lasting microglial reprogramming that we can suppress pro-inflammatory gene expression with repeated LPS. We then wanted to know what's the functional consequence of this. And so we looked at one of the key functions that microglia do in the brain. So they phagocytose invaders, and they also actually phagocytose synapses. This happens naturally during early development, but can also happen in the context of chronic disease conditions. And so we developed a flow cytometry assay to look at microglia that had engulfed synapses, so we can stain for synaptic contents inside microglia. And we predicted that we would see changes either in the number of microglia or in the percentage percentage of positive microglia or in the amount of microglia that were positive. And so that's exactly what we found. So we could look at the change in proportion of microglia that were positive for vGlut1, which is a marker of excitatory synapses. And as you can see here, we did not see any changes in the controls. We saw an increase in phagocytosis in the acute LPS that was suppressed by the repeated LPS. The next thing that we wanted to do was take a look at what the actual microglia look like. And so this was done by my graduate student, Jennifer Kim, who developed a new tool to look at changes in microglia morphology. So I already mentioned during inflammation, microglia take on this amoeboid or rounded shape, but there are actually many different types of morphology that microglia can have. So they can have these hypertrophic microglia, which we see in stress conditions, rod-like microglia, which are sort of long, thin ones, or these homeostatic ramified microglia. And she developed this neat tool to actually quantify this in thousands of microglia across the mouse brain from immunohistochemistry images. We take a total of like 27 different metrics of the microglia, things like how branched they are, how tightly packed they are, And then this all gets fed into a PCA analysis where we can then use the PCs to cluster the microglia into individual states. And if you're interested in this, this is actually now published in eNeuro, and the code is all open source and easily accessible. Jen made a very nice YouTube video to help walk people through this, and our images are actually also available, so you can play with them if you'd like, or use some of your own. So what actually happened? So this is an example of IBA1 staining. So this marks mostly microglia in the brain. We were then able to identify these different microglial states using Jen's new tool. And then we were able to quantify the percentage of microglia in each one of these different states across our 4 different conditions. And just to give you the take-home message, we see the most changes in these amoeboid microglia, which are the pro-inflammatory ones. We see an increase after LPS that lasts for 7 days. So this is a bit different than the gene expression. This goes up even higher acutely in response to LPS, and this is blunted in response to the 2x LPS, indicating that our tolerance phenotype is able to extend both to phagocytosis and also to the actual shape of the microglia. So in conclusion, what I wanted you to take home today is that LPS blends sickness behavior to a subsequent LPS injection. We're also able to modulate pro-inflammatory gene expression in our tolerance paradigm, and this has long-term impacts on phagocytosis and changes in morphology. But of course, this is the beginning of a much larger ongoing project. We're really interested in what the actual epigenetic mechanisms might be that control these changes in gene expression. And while LPS is a great model, we're interested in also expanding this to other types of models that are more physiologically relevant for different disease states. And so, of course, none of this could have been done without all the wonderful folks in my lab. I talked about work from Morgan and Jen, but we have many other projects ongoing in the lab. And of course, none of this would be possible without the support of all of our collaborators and our funding agencies. And so with that, I'd like to say thank you for attending this webinar, and I look forward to your questions.
Thank you very much. So, questions we have. Well, I'll ask a couple of questions first. First, oh dear, hold on. First of all, to Raul. I just wanted to ask you, Raul, what in your 2D versus your 3D cultures, what's the percentage of microglia to astrocytes to oligos to neurons, particularly in the 3D?
We haven't quantified as such the number, but what we see from experience from working with the 3D culture is the most abundant cell type is the astrocytes. Then they are followed by neurons, and then microglia are kind of, I would say, 10% of the culture, and then around 10% of oligodendrocytes. The only thing is that oligodendrocytes they mature later, so they take longer to reach a phenotype. So that was a little bit difficult to work with.
How old are your cultures then? How old are your cultures?
So for the results that I showed you, they were 12 days in differentiation. However, for the oligodendrocyte studies, we were able to bring them up to 25 days in culture after day 1 in vitro, after the establishment of the culture. And the interesting thing about the matrigel is that it kind of holds the astrocytes from dividing, because when you have a plate, because you can do a co-culture in 2D, right? Then you just put a bed of matrigel or poly-D-lysine, and that will hold at least neurons and astrocytes. But what happens there is that astrocytes usually tend to overcome the neurons, and in the end, you find very few neurons, and most of it is just astroglia. But in this one, the little sphere of matrigel seems to hold the division, and we do not see we do not see this overcoming of the neurons. Actually, they start to correlate, as I showed in my last slide, where you can see how astrocytes start to cover connections.
Okay,
of
great.
the
We have a question here. Do you see changes in oligodendrocytes?
Yes, I didn't show you the data, but we did publish a paper about that. And what was strange is that 27OH was speeding up the differentiation of the oligodendrocyte precursor cells. So OPCs were expressing earlier maturity genes compared to the controls. And we are still trying to figure out what it means physiologically. We think that this is a way for the oligodendrocytes to try to cope with the abundance of 27OH that they might be misinterpreting as a lot of cholesterol around them.
Okay, great. This is a question for Annie. What would happen if you treated with LPS every day for 7 days or longer? Yeah, we did this for stress.
Yeah, great question. So we've done some of this at a slightly lower dose. So we have a paper that just came out looking at 4 days of LPS injections at like a much lower dose, and you also can produce tolerance. We haven't done the higher dose of LPS for multiple days and looked at some of these same measures yet. The animals get fairly sick, so I don't know how well they would survive or do if we kept giving them higher doses of LPS repeatedly. But yeah, it's interesting because the rules around dose and timing and what the stimulation is and how that impacts the microglia are not really very well worked out. Like lots of people are using LPS, but it varies widely in the doses and the types of effects people see.
Yeah, yeah. How old are your animals? I mean, what's the age of your animals?
Yeah, these are all young adults. We do have some work also looking in much younger animals like early postnatal. And again, the dose changes dramatically. Like if you gave them an adult dose, they'd die. So we scale things back, but we are looking at some postnatal time points too. But yeah, all of the work I showed today was an adult.
Okay. Another question for you, Annie. Thank you for a brilliant presentation. Do you see the microglia taking on different expression profiles when they've got tolerance, e.g. antigen presentation versus phagocytosis?
Oh, good question. We have not looked at antigen presentation, which is a bit controversial in microglia in the brain, in at least the homeostatic brain. So in the context of like multiple sclerosis, you can see them presenting, although they're thought to be very inefficient at presenting antigens, and it's not clear who they're presenting to. In this context, I could look through our gene list. We did not get any like antigen presenting related genes as our top hits. But again, we're sorting for microglia too, so it's possible other cell types might be doing that instead. Yeah. We are starting now to look at peripheral immune cells that might be infiltrating in response to LPS.
Yeah, I just wondered, have you any indication about what's happening with the blood-brain barrier when you do your multiple doses?
Yeah, we don't know in this paradigm. That's something that we're actively working on. The microglia that we isolated for sorting were done with P2YR12, so they are microglia. They're not infiltrating cells. We've looked by IBA1 staining, which can pick up infiltrating cells and microglia, and we don't see changes in the total number of those cells in our condition. So if there is infiltration, they're either coming in transiently or there's not a huge number of them, or they're not IBA1 positive. So we have some flow cytometry assays to look.
So you're treating systemically with LPS?
Mm-hmm.
So what about things like the influence of cytokines from peripheral immune cells on the
great question. Yeah, I didn't show that data, but the peripheral immune cells, so we've isolated PBMCs from these same animals, and they do not show memory. So they respond to every LPS dose as if it's the first one, and it's probably because in that like 7 days, some of them are turning over, especially in that 30-day time point, they'll have been replaced. If we looked in the bone marrow, maybe we would see some memory phenotypes, but yeah, we don't think that the brain microglia changes are being driven by the periphery because we just don't see changes there.
Okay, we have a couple more questions. Do you think astrocyte microglial interplay determines influences phenotype of each cell type in neurodegenerative diseases?
So is
To me,
this open?
It could be, yeah, I think we could say, yeah, it does, you know. Thank you, Annie, for a great presentation. Do you plan to or have any other studies examining the effects of maternal infection, i.e. LPS exposure of pregnant females on microglial tolerance in the offspring?
Yeah, great question. There's been a lot of work done on this, and I will point people to Lindsay Hayes's work. She did some of this in her postdoc, really elegant work, and she now has her own lab where she's doing these types of like in utero manipulations. I did some of this as a postdoc, and it's a lot harder to control than it is to do it just in the adult, where you know exactly the dose each animal gets. So we started in the adult, but I would like to go back to like some of those maternal immune activation models at some point.
I suppose you can't really mimic it that easily, but also that is how you would get changes. I mean, we've done stuff with stem cells, human stem cells, where you stress them, take away the stress, and you get changes in the microglia at the end of it. So
and I mean,
you'll definitely see changes, epigenetic changes.
Yeah, and microglia come from this unique cell type in the brain. They come from a progenitor population in the yolk sac primarily, and they enter into the brain during embryonic development in mice and humans. So they have a completely different cell lineage than the rest of the cells in the brain. So yeah, the epigenetic changes that happen to them and their selective vulnerability is something people are certainly interested in trying to understand.
Yeah. Okay, well, I think that's actually, I've just got one more question for you, Raul. Do you know whether your astrocytes mimic type 1 or type 2?
Yeah, that's a very interesting question. We don't know. At the time when we were doing this research, and at the time that the first paper came out from Sten Linnarsson, where they were sequencing astrocytes, and they identified lactaderine as a marker for the type 2 astrocyte. So we did started to do an immunohistochemistry for lactaderine to try to see in the brain of the mouse if we had a change in the population. However, that didn't work out very well because there were not very good antibodies at the time for lactaderine, and we had staining on neurons. So then, yeah, then we didn't continue that avenue. So that was a big thing that I remained to study. Yeah. I do believe that there might be a change in the population.
Quite nice to find out if that's true. Right. Well, thank you both. Well, we had the questions. Thank you for attending, everybody attending, and thank you very much to the speakers. Great talks, and hopefully continue with this research. You can continue the conversation online, following at BiochemSoc and at PP Publishing on X. It says Twitter, but it's not anymore, is it? We welcome suggestions for future topics and speakers to feature in this biochemistry focused webinar series. If you have any ideas, please let us know and propose for any upcoming webinar. You can find more information about webinars at www.biochemistry.org. All our upcoming webinars are listed on our website as well, and if you've missed any, you can find them there because they're part of the series. This webinar will be on YouTube and the website. Sorry, what is going? And this webinar will be available in actually a few days' time, and it will be posted on the website. So thank you, everybody, again.
Thank you.
Yeah, thank you.
Yeah, thank you. Bye for now. Oh, delete the survey.